Good day, students. Welcome on behalf of Science Academia. Today we will read about a very short topic yet interesting, which is about the lysosome, which is a cell organelle. So let's start. This is the diagram of a simple lysosome. Lysosome structures are definitely they are the most simpler ones, mostly spherical. They range in size from 0.2 to 0.5 micrometer as mentioned below, and the internal pH which they maintain is highly acidic and it is maintained at approximately pH 5, around 4 to 5. They are surrounded by a membrane, and all the hydrolytic enzymes whatever are present which helps in intracellular digestion of different foreign particles or self particles are present within the lysosome, either in active form or in inactive form. These different pigments are in the figure. The different hydrolytic enzymes are depicted in short blue colored compounds, red colored compound, pink colored compound, as you can see within the membrane of the lysosomal structure. Okay, so this is all about the structure. Now let's discuss about the features of lysosome. Now lysosomes are single membrane bound organelle present in almost all eukaryotic cells with exceptions. There are few exceptions like euglena, few protista like euglena, etc. Those exceptions are there. Exceptions are there almost everywhere, but if we overdo those exceptions, then almost all eukaryotic cells, almost 97% of them contain lysosomes. Okay, they have an acidic pH which is around 4.5 to 5 and contains nearly 40 different hydrolytic enzymes. So can you see the variety? How many hydrolytic enzymes they contain? They contain about 40 different kind of enzymes which can be grouped into 5 categories, which we will discuss in these slides in later parts. Now the third point says the enzymes are acidic in nature and hence the lysozyme provides the best environment for their optimal activity. Obviously the enzymes are acidic in nature, so they are able to degrade the substances, and if the lysozyme or the lysosome doesn't provide them the optimum acidic temperature, then how will they survive? If the lysosome was basic in nature, then they would have been degraded or they would have been totally destroyed. If it would have been neutral, they would have been totally in inactive form. So the condition which is most favorable for their optimal activity is acidic, and lysosome provides that acidic nature or temperature. Okay, now in association with this I have asked you a question in the end of the slide. Let's look at it first, then we will discuss all of these. So how does lysosome maintain its acidity? The enzymes present are acidic in nature. To supply them the optimal activity, the lysosome is acidic in nature. Okay, but the question is, how does the lysosome maintain its acidity? Well, this answer I will post in our Instagram page today, so you can check our Instagram handle and see the answer, or if you know, you can write it in the comment box below. Okay, so please check our Instagram page. It has the name Science Academia. I will give it in the description box, the link. Okay, now let's come back to our lecture. Okay, so we discussed about the features. Now we will discuss about its type. So lysosomes are basically of two types, which are primary lysosomes and secondary lysosome. Now they can be differentiated in a very subtle and soft way with only one basic difference. In simple words, just to say that primary lysosomes contain hydrolytic enzymes in inactive form, and the secondary lysosomes contain them in active form. So when the enzymes are present in inactive form, it is very obvious that they do not help in digestion of any foreign or self material, while in secondary lysosomes, as the enzymes are activated, so they can help in undergoing digestion. So now let's read what is written here. Inactive hydrolytic enzymes, as I told you, are present or do not contain any material for intracellular digestion. This material only means enzymes. Understood? And what about secondary lysosomes? Contain materials that are undergoing digestion or already digested. So these are the two forms, primary and secondary. We also have the tertiary and the quaternary forms, or the actually quaternary or the autophagosome or lysosome. Tertiary form is what, you know, after digestion the residual bodies which are present within the lysosome, that particular structure is termed as the tertiary structure. And autophagosomes or autolysosomes we will study in next slide. So there are four types, but the main primary division is primary and secondary lysosome. Number one is primary and number two is secondary. Okay students, now let us divide lysosome according to its digestive power. Okay, so lysosomal digestions can be classified into two types. One is autophagy and one is heterophagy. So what does autophagy means? From the name only we can understand that this is related to something self, auto, and hetero is something different from taken from outside. So what does autophagy means? It means digestion of cytoplasmic components within lysosomes. Like when the cell signals that there is some defect within its any of its organelle, like maybe the mitochondria is not working, or any of the Golgi apparatus or something like that is malfunctioning, it has to be removed or it is of no use, then the cell provides the signal and lysosomes present within the cell gets activated and they digest these cytoplasmic components. So this is known as autophagy. Okay, and this particular process I was talking about, autophagosomes or autolysosomes, these autophagosomes are associated with autophagy. So as they use their own material, this does not happens in all normal cases. Our cells do not destroy its organelles. Whenever there is a need to do so, they do so. So in those particular cases, what they do, the cell particular organelles are destroyed and the cell is lysed. So these, and at that particular case lysosomes are termed as suicidal bags. Okay, and what about heterophagy? In heterophagy what happens? The lysosomes engulf the substances from outside or the foreign particles, any kind of foreign particle, maybe food particle or any kind of pathogen which are ingested and then followed by digestion within themselves. Then this type of digestion leads to heterophagous lysosomes. So you understood, according to digestion they are differentiated into autophagy and heterophagy, and according to their functional role they are differentiated into primary and secondary as their functional role of enzymes. Now let's see the third division, in which we will differentiate heterophagy on the basis of their two types. The first type is phagocytosis and the second type is pinocytosis. In phagocytosis what happens na, the cell engulfs large particles. So phagocytic cells you have studied, if not then we will study further. Just for now you know the names like macrophages and neutrophils. These are phagocytic cells. What they do, they engulf large foreign particles, and then after engulfing these large foreign particles they create a circular structure, a vesicle kind of structure which is known as phagosome, which then fuse with the lysosome to form the phagolysosome, and further then these foreign particles are degraded within the phagolysosome. This is the process of phagocytosis. And what about pinocytosis? Approx all eukaryotic cells internalize the fluid substances in small pinocytic vesicles. So there are small vesicles which happen to take birth within the territory of the cell. Okay, and these vesicles, what they do now, they take up small substances, very small substances, not like those in case of phagocyte. They used to engulf large particles. Here very small substances which may be foreign, food particle, any pathogen, they are taken in the form of small vesicles, and this particular process is only known as pinocytosis. And what happens after they are engulfed, they eventually bud off inside the cell as a independent circular or maybe any shaped, but as an independent structure they bud off, and at this particular stage they are known as early endosome. Now the early endosomes, what happen, they differentiate to form the late endosomes. By rapid division they increase in number, so by pinocytosis they bud off inside and form a structure which further mature to form early endosomes, which further differentiate and mature to form the late endosome. Now the process is similar to what happens with phagosomes. They fuse with the lysosome. Here also they fuse with the lysosome, these endosomes, and to form the endolysosome, and then they are degraded by the hydrolytic enzymes. So the end process is same in both the cases. We will see in the diagram. Okay. And autophagy I have already discussed, but here also I will read it for you once again. Phagophores. Now what are phagophores? Those molecules only I was talking about now, like for example, if a mitochondria in a cell is very much destroyed or not active or not needed, which is causing very much disturbance in the cell, which is totally non-functional, so that parts need to be emitted, needs to be destroyed. So those particular then organelles, what happen now, they take up the structures of small tiny, they disintegrate and they form phagophores, small tiny porous like structures, and they together form the phagophores, which then fuse with the lysosome to form the autophagosomes, and then further they are fused with the lysosome to degrade their internal material, and this is only known as the suicide thing of lysosome. You understood now? Okay, this diagram will make it clear if you have not understood. So see, the first channel is of the phagocytosis process, in which you can see that a foreign particle, large foreign particle, imagine this cell to be a neutrophil or a macrophage for this particular phagocytosis process. Now this star shaped material is a foreign particle, see it is engulfed and a phagosome is formed. Then the phagosome fuses with the lysosome and further they are degraded using the hydrolytic enzyme. This is process number 1. Everything is happening within the cytosol. Okay. Now in the second process is pinocytosis. See, small particles are engulfed, therefore through the process of pinocytosis, then they form the early endosome, which differentiate and form a greater structure, matured structure, which is the late endosome, which then further fuse with the lysosome to form the endolysosome, and then they are further degraded by the lysosome. And the last process is the autophagy or the suicidal process, in which the phagophores, see the phagophores here, the small particles which are to be digested, are totally enlocked within a double membranous structure. Okay, what happens now, most of these phagosomal materials, what happened, these phagophores are formed de novo in the cytoplasm from a cup shaped membrane. De novo means a new pathway. So these phagophores are formed, and these cup shaped thing, see, this only takes the structure of this cup shaped thing, cup shaped membrane that expands into a double membrane structure. See, they have expanded into a double membrane structure, and they form the autophagosome with the foreign material inside of them. These autophagosomes may fuse with the endosome, so they may take the other pathway, they may fuse with the endosome to form the amphizome. What? Amphizome. AMPHISOME. See, I have mentioned here. If they fuse with the endosome, then they may form the amphizome. Or else they may not fuse, and directly they may fuse with the lysosome. In that case, they will be degraded. If they fuse with the endosome to form the amphisome, and then they will fuse with the lysosome, in that case also they will be degraded. So the resulting macromolecules which are degraded are released through the cell outside and are recycled into the cytosol. So in this cytosolic structure only they are released and they are again recycled. And if these are waste material which needs to be excreted from the cell, then through various transport processes they are excreted out of the cell. So these are the three basic processes of lysosomic degradation: phagocytosis, pinocytosis and autophagy. Okay, so this is all about the functions of lysosome. Now I told you earlier that there are about 40 different kind of hydrolytic enzymes. So the different types of hydrolytic enzymes can be grouped into 5 categories. Okay, so one is phosphatase, one is nuclease, one is polysaccharide hydrolyzing enzyme, polysaccharides or mucopolysaccharides. The other one is proteases and the other one is lipid degrading enzymes. There are many examples in between them. I have taken two. You can read these two only, and if you can memorize or remember the other ones, you can take them from book or internet, whichever is available to you. So the examples of phosphatase are acid phosphatases and acid phosphodiesterase. In case of nucleases, which are mainly DNAs, RNAs, so these are acid ribonucleases or deoxyribonucleases. In case of polysaccharide hydrolyzing enzyme, we have hyaluronidase or beta galactosidase. In case of protease, we have collagenase and peptidase. And in case of lipid degrading enzyme, we have esterase and phospholipase. So this is all what I had to taught you in the lysosome part of our lecture. Do answer this question in the comment section if you know, or else you can visit our Instagram handle Science Academia and see the answer. Okay, so thank you class. If you like our lecture, if you like our content, please do subscribe to our channel, like this video, share it with your friends so that they can also gain this kind of knowledge. We are a new setup and we are working hard for you people. So please do subscribe to our channel and do show us some support. Thank you people. Be safe in this situation of pandemic. Okay, study in home and study well and be positive. Thank you so much.